Hi students, how are you? This is the first video that I am attempting to do in order to explain the different living structures that we need to see for finishing this third partial of biology number 1. We are going to start with the explanation of viruses. To do that, you should remember that viruses are not living things, or they're not considered living things. Why? Because they are not formed by cells. They are not formed by cells, and despite of their very, very complex structure, they look very, very complex, they are not formed by cells. They are only formed by two different elements: an external protein case known as capsid, and DNA or RNA that can be contained in the interior. For example, in this virus, we have an external case made by proteins. All this structure will be protein, and inside we will have the DNA. Remember, just as we saw it. The same is going to happen with this example: an external case of proteins, and inside DNA. They don't have membranes, they don't have organelles, and beside that, they do not possess any of the characteristics of living things. In the world, we can find many different types of viruses, many different shapes, and many different cells or organisms that they can infect. But one of the important things about understanding viruses is that generally they receive the name of the disease they produce. For example, we have the rabies virus, influenza virus, also very, very known, very, very famous influenza virus. Also, we have HIV. This is the virus that produces AIDS. And well, there are many other types of viruses. But what will happen if we try to compare a viral structure or a viral particle with an actual cell? Well, this picture will be very helpful for that purpose. For example, the brown thing that we are seeing over here is just a comparison or a size of an actual eukaryotic cell, like the one that has our body. Remember, eukaryotic cells are characterized because they have a nucleus. Now, this structure over here is a prokaryotic cell. We compare the size of a prokaryotic cell with an eukaryotic cell, and it is just smaller. Remember, prokaryotic cells are just the ones that forms bacteria. And finally, to complete this exercise of comparison, the virus will be this just tiny dot. So virus at the end are very, very small particles. Remember, again, they are not cells. They are very small particles that can infect also bacteria and also eukaryotic cells. Actually, this case over here, bacteriophage. Bacteriophage is a type of virus that infects particularly bacteria. As I said earlier, viruses, because they are not made by cells, will not have any other characteristic of life. They do not have metabolism, they don't have growth, neither development, they have no irritability. The only property they can have is just a sort of reproduction, but we can call it as an actual reproduction. We should better call it as replication. You're going to see that a little bit further. Well, another important thing to know is that this replication or reproduction can be divided in two different ways or two different cycles. just in viruses: lytic cycle and lysogenic cycle. For a virus to replicate, or in this case, reproduce, remember the actual word or the actual concept will be viral replication, the first step must be the virus entering to the cell, entering touch to the cell, or getting very close to the cell. For doing that, you should remember that all the cell membranes have in their external structure some sort of proteins that we call it as receptor proteins that are in charge of recognizing other cells, or in this case they allowed viruses to attach directly to this site. They are like docking sites where the virus can attach to the cell. The important thing about these sites is that they are specific. For example, in the case of this virus, it will infect only the cells that have this particular type of receptors. In other words, an influenza virus will never affect liver cells or muscle cells. Influenza virus have the ability to recognize and attach only to the receptors of respiratory cells. Step number 2 will be virus duplicating itself inside the cell. But for doing that, previously the virus must inject or insert the genetic material into the cell. This is an example of HIV infection. Remember, HIV is a virus that produces AIDS. Once again, we have the first step that is also known as docking, or just recognizing the viral particle with the cell membrane, the host cell membrane. For doing that, we have the special receptors. Number 2, the second step, will be the virus injecting or inserting the genetic material into the host cell. In this case, HIV virus does not have DNA as genetic material. It has RNA. Once the genetic material is inserted inside the cell, the third step will be that this genetic material will be fusioned or mixed with the host cell. For doing that, in this case, viral RNA must suffer a process of reverse transcription, a process that we are not going to explain right away, but you should remember there is a way for the cell to change from RNA into DNA. Once the DNA is contained inside the cell will use it as a blueprint for creating new viral particles. So, step 1, virus entering the cell, virus injecting or inserting its genetic material inside the cell. Step 2 will be the virus replicating itself or duplicating the different parts. And finally, the expulsion of all the new viral particles. Now, as you should remember, previously I said that the viral replication or viral reproduction in this case can go 2 different ways. One is known as the lysogenic cycle. This is when the genetic material of the virus gets mixed or fusioned with the host cell. In this case, you can see the viral particle already mixed with the host DNA. This is known as lysogenic cycle. The other option will be known as lytic cycle. Once the genetic material is mixed, this is going to be used as a blueprint for creating the new parts of new viral particles. Once the new viral particles are formed, generally the host cell will explode or will be destroyed for releasing the viral particles outside. This is very important because at the first parts or the first stages of a viral infection, we're talking about lysogenic cycle, we will not have any symptom of the disease because the genetic material of the virus, the viral DNA, is mixing with the host DNA. When we're going to start feeling the symptoms will be when the cells break or are destroyed by the expulsion of the viral particle in lytic cell. This part of the cycle is one of the most dangerous of all because the host cell is destroyed by the action of the virus. Once again, only remember that viruses are not made by cells. They are only formed by an external case of proteins known as capsid, and inside they contain DNA or RNA. They will not possess any characteristic of living things as metabolism, growth, reproduction, irritability, besides reproduction. The process of reproduction can be divided in two: lytic and lysogenic reproduction. The one that absorbs DNA and make it itself as part of the host cell is known as lysogenic, while the one that destroys the host cell is the lytic cycle. Well, I hope that this video can be useful for you. Remember, we're going to start seeing a series of different videos to try to understand the different biological structures that we can find in the world. Well, see you later.